Hello friends, today we will discuss about an important topic in biochemistry. You will get short note on individual RNAs and this is an important LAQ in university examination. Ribonucleic acid. Ribonucleic acid. As compared to DNA, it is a single-stranded molecule. Single-stranded molecule. In some places, due to the complementarity between the bases, it appears like double-stranded molecule. Ribonucleic acid, it is a polymer of ribonucleotide. Ribonucleotides, or it is a polyribonucleotides connected by 3' 5' phosphodiester linkage. 3' 5' phosphodiester linkage. We know the concept of nucleoside and nucleotide. This is a ribose sugar. To this ribose sugar, there is phosphate attached. This is a 5' end. And this is 3' carbon. This is first, second, third. To this 3' carbon, there is attachment of phosphate of incoming nucleotide. This is another ribonucleotide. This is the first one, this is the second one. To this there is attachment. Suppose this is adenine and this is uracil. This one is 3' 5' phosphodiester linkage. 3' 5' phosphodiester linkage. This one is another nucleotide. This is the first nucleotide, adenine. The nucleoside of adenine, it is known as, this is nitrogen base. Adenine is the nitrogen base. The nucleoside of adenine is known as adenosine. Adenosine. The nucleoside of uracil, it is known as uridine. It is known as uridine. The nucleoside of guanine, it is known as guanosine. And another base which is found in RNA, it is cytosine. The nucleoside of cytosine is known as cytidine. These are nucleoside. Nucleoside means the ribose sugar plus nitrogen base. Ribose plus nitrogen base, it is known as nucleoside. If we add the phosphate, it is known as nucleotide. The nucleotide of adenosine, it is known as adenylate. Adenylate, or another name is 5' adenosine monophosphate. Guanosine, it is guanylate. Cytidine, it is cytidylate. Uridine, it is uridylate, or it is known as 5' uridine monophosphate. Got it? Adenosine, adenylate, guanosine, guanylate, cytidine, cytidylate, uridine, uridylate. These are nucleoside, means ribose plus nitrogen base. These are the phosphate, means adenylate, guanylate, cytidylate and uridylate. Another name is 5' AMP, 5' GMP, 5' CMP and 5' UMP. This is RNA molecules, single-stranded polyribonucleotides and 3' 5' phosphodiester linkage. There are three major RNA molecules, that is mRNA, rRNA and tRNA. Messenger RNA, ribosomal RNA and transfer RNA. Okay, these are the three major types of RNA. from this DNA molecule, suppose this is a DNA molecule, from this DNA molecule, by the action of polymerase 1, RNA polymerase 1 will get ribosomal RNA. From RNA polymerase 2 will get messenger RNA, and from RNA polymerase 3 will get transfer RNA. So these are the three major types of RNAs. Now we'll see the one by one: messenger RNA, transfer RNA, and ribosomal RNA. First we will discuss mRNA. This M, it is for messenger. This mRNA carries the message from DNA to ribosomes or protein synthesizing machinery. It carries message from DNA, or it carries message from or genetic information from DNA to the site of protein synthesis, that is ribosome. This is a prime function of mRNA. The precursor molecule of mRNA is known as hnRNA, heterogeneous nuclear RNA. It is a precursor of mRNA. This mRNA, it is synthesized by RNA polymerase 2 in eukaryote. RNA polymerase 2 in eukaryotes. First point is it carries information from DNA to the site of protein synthesis. Second, it is from the precursor molecule, that is hnRNA, heterogeneous nuclear RNA. It is synthesized by RNA polymerase 2, and it constitutes about 5 to 10% of total RNA. Now in structure, this mRNA, it is composed of 5 parts. It is composed of 5 parts. At the 5 prime end, there is cap. There is cap. At the 5 prime end, this cap is of 7-methyl guanosine triphosphate. At the 5 prime end, there is cap. It is of 7-methyl guanosine triphosphate, which is connected by 5 prime triphosphate linkage. Okay, so there is 7-methyl guanosine triphosphate cap at the 5 prime end, followed by untranslated region. Which is known as 5 prime untranslated region of mRNA. Then coding region. This is coding region, followed by 3 prime untranslated region. This 3 prime untranslated region, it is followed by polyadenylate tailing. Okay, so these are the 5 regions in mRNA. First one is the cap, second one is 5 prime untranslated region, next one is the coding region, then 3 prime untranslated region, and poly-A tail. This 5 prime cap of 7-methyl guanosine triphosphate, it prevents from the exonuclease attack. It prevents from 5 prime exonuclease attack. Second, it promotes the binding of mRNA to the ribosomes, ribosomal binding, and it gives stability to mRNA, and it is important in transfer of this mRNA from nucleus to cytoplasm. So these are the important function of this cap. Then this coding region, it is divided into 3 parts. First one is the initiation codon, that is AUG. the triplet base sequence which is present, or triplet nucleotide which is present on mRNA, it is known as codon. This AUG, it is initiation codon, followed by specific codons which codes for different amino acids, and stop codons. It includes UAA, UGA and UAG. These are the three stop codons which stop the process of translation. So this is about the mRNA. In eukaryote, this mRNA is monocistronic. Means they code for single polypeptide. This is in contrast to prokaryotes, in which the mRNA is polycistronic. In eukaryote, it is monocistronic. They code for single polypeptide. That's why they are known as monocistronic. So this is all about mRNA. It is known as messenger RNA. It is important in transfer of genetic information from DNA to the site of protein synthesis. The precursor molecule is heterogeneous nuclear RNA. After the post-transcriptional modification, this hnRNA is converted into mRNA. mRNA has 5 parts, that is cap, untranslated region, coding region, 3' untranslated region, and poly A tail. This poly A tail prevents from the attack by 3' exonuclease. It constitutes about 5 to 10% of total RNA. And this mRNA, they are also known as coding RNAs. All other RNAs, they are known as non-coding RNAs. This is all about mRNA. Now we will discuss the tRNA. The important, in university exam, you will get a short note on tRNA. It is a very, very important SAQ. tRNA, it is a very, very important SAQ. tRNA. T means transfer RNA. It is transfer RNA. It is also known as soluble RNA. They are small molecule. The molecular weight is 25,000. The structure of tRNA, it is given by Hawley. The secondary structure of tRNA, it is known as cloverleaf model. While tertiary structure, it is L-shaped. And it is important, it get accommodate in the A site, that is amino acid site, and peptidyl site of ribosome. This three-dimensional L-shaped tertiary structure of tRNA. It get accommodate in the amino acid site and peptidyl site of ribosome. That's why it is important. 3D structure of tRNA. Secondary structure, it is known as cloverleaf model. Near about 75 to 90 nucleotides are there in tRNA structure. There are unusual bases in tRNA. For example, dihydrouracil, it is an unusual base in tRNA. Then thymine, that is ribothyamine, it is an unusual base. Then methyl adenine and dimethyl guanine, hypoxanthine. These are the unusual bases in tRNA. Now we will discuss the structure of tRNA. The important function of tRNA is to transport or transfer amino acid to the ribosomes. in structure, remember it is from 5 prime end to 3 prime end. The sequence is DATA, data. This is the sequence to remember tRNA structure, DATA. Okay, this is 5 prime end. This is first is the D loop, followed by anticodon. This is variable arm. This is T psi C loop, and this is uncomplementary region, that is CCA. This is a 3 prime end of tRNA. Okay, this is data D anticodon, DA T psi C loop, and this is acceptor arm. Acceptor arm. So these are the 4 important part. This is a variable loop. Okay, this is 5 prime end. Then it has from this DATA, this has double stranded complementary region in this arm. This D arm, it has 4 to 6 nucleotides. It is 4, 5, 5, 7 nucleotides. It has 4 nucleotides complementary. Then this anticodon, it has 5 to 7. Then 5 and is 7. And this is uncomplementary region, that is CCA. At this end there is attachment of amino acid, and on the basis of attached amino acid, this tRNA, they are classified into 20 types. Five prime end, D arm, anticodon loop, T psi C arm, and acceptor arm. This D, it is because of dihydrouracil, which is an unusual base present in the tRNA. Then T psi C, T means thymine pseudouridine, and cytidine arm. Thymine, pseudouridine, and cytidine. This is known as pseudouridine because the linkage, the glycosidic linkage between the ribose and uracil, it is in between carbon C1 of ribose and C5 of uracil. Usually it is between C1 and N1 of pyrimidine, C1 of sugar and N1 of pyrimidine, but in this case it is between carbon and carbon. That's why it is known as pseudouridine. This loop is known as D loop because of dihydrouracil base, which is an unusual base present in the D arm. And this is anticodon region, 1, 2, 3. Anticodon region, which attached with the codons of mRNA, and after this attachment there is decoding of information occurs. So transfer RNA, it is responsible for transfer of amino acid to the site of protein synthesis, that is ribosomes. So this is about the tRNA. Transfer RNA, soluble RNA described by Hawley. Then it has near about 75 to 90 nucleotides. It is a smaller molecule, has molecular weight 25,000. These are the complementary bases which are present in this loop: D arm, anticodon, T psi C, and acceptor 4, 5, 5, 7. Okay. This is a anticodon arm. It helps in the attachment to the codons of mRNA. That's why it is known as anticodon arm. And this A acceptor arm, the amino acid get attached to the hydroxyl group of this CCA end. So this is all about the structure of tRNA. Now we'll see the structure of ribosomal RNA. These tRNA, they constitute about 10 to 20% of total RNA molecule. Now we'll see the structure of rRNA, ribosomal RNA. Ribosomal RNA, they are also known as catalytic RNAs or ribozymes. Ribosomal RNA, it constitutes about 60 to 80% of total cellular RNAs. They are also known as catalytic RNAs. They help in the synthesis of ribosomes. They help in the synthesis of ribosomes. Ribosomes, they are made up of RNA, rRNA plus proteins. Ribosomes, they are made up of rRNA and proteins, and these ribosomes, they are studied in the endoplasmic reticulum, and it is known as rough endoplasmic reticulum. Also known as catalytic RNA or ribozymes. You may get a short note on ribozymes. Now in eukaryote and in prokaryote. In prokaryotes, the ribosome is of 70S unit. This is ribosome. It is of 70S unit, that is Svedberg unit, which is dependent on molecular weight and size. Molecular weight and size. It is of 70S, which is dissociated into 50S ribosomal unit and 30S ribosomal unit. This 50S ribosomal unit, it is made up of 23S RNA, 5S RNA, and near about 34 proteins. While this 30S subunit, it is made up of 16S RNA and 21 proteins. This is about prokaryote. 70S ribosome, it is dissociated into 50 and 30S, it is Svedberg unit. Okay. And this 50S, it is made up of 23S and 5S RNA and near about 34 proteins. Near about 34 proteins. It is made up of 16S RNA and near about 21 proteins. While in eukaryotes, it is 80S unit. It is of 80S unit, which dissociates into 60S and 40S ribosomal unit. This 60S ribosomal unit, it is made up of 28S, 5.8S and 5S RNAs plus near about 50 proteins. And this 40S subunit, it is made up of 18S RNA and near about 35 proteins. This is ribosomal RNA. It constitutes about 60 to 80% of total RNA. And this main important function of these ribosomes, it is a protein synthesizing machinery. It is a site for protein synthesis. site for protein synthesis. Also, it helps in the formation of peptide linkages. It helps in the formation of peptide bonds in the polypeptide chain formation. Help in the synthesis of peptide bonds in the polypeptide synthesis. This is an important function of ribosomes. So this is all about mRNA, tRNA and ribosomal RNA. Apart from these three major types, there are also small nuclear RNAs, which helps in splicing of mRNA or processing of mRNA. They are known as SNRs, small nuclear RNAs. They are pronounced as SNR. They help in splicing or processing of mRNA molecule. So this is about the different types of RNAs. Keep watching. Thank you.